just give you a little orientation. Let me just give you a little orientation to where we're going. Before we go anywhere at all, I want to thank ISCP and particularly Eduardo. Eduardo, thank you very much for inviting me to give a talk, and I appreciate you guys investing a little bit of time in hearing the story that I have to share today. And we're going to start out by talking about a dissociation or several ways of dissociating active working memory from passive familiarity signals in rhesus monkeys. That's work we've been doing for a decade or so in my lab. And then we're going to take that as a background and talk about some work we've been doing that's probably in its early stages, efforts to compare these memory processes that we dissociate in monkeys across non-human primate species. And that quickly evolves into an apparent mystery about memory in orangutans that I'll share with you. And Eduardo said we will have a little time for questions, and I'd love to hear people's thoughts about what's going on in this mystery. We are openly calling it a mystery. I'm a bit puzzled by these outcomes, and I'm interested to hear if people can make much of it. So let's just start out by setting a real basic backdrop that most of you are going to be familiar with, matching to sample. It's a technique that we use a great deal in my lab and in other labs, of course. And in a matching to sample paradigm, what you do is all the things I'm going to show you are an animal sitting in front of a computer screen. So they see a picture at the beginning of a trial, and they study that, and they tell us they've studied it by touching it a couple of times with their finger. Then there's a delay period, and during that delay period, the animal has to remember what it saw. And at test, it's going to get presented with the picture it saw at the beginning of the trial, and several distractors that it's not supposed to choose. If it chooses the one that it saw at the beginning of the trial, in this case, picking the rooster again, by touching the bottom left, it can get a food reward, which might be a peanut or a primate pellet. I'll give you just a quick little video of a monkey in our lab doing one of these tests. In this case, this monkey is doing a face matching task, which that's another story. I'd be happy to talk to anybody about that once. It turns out they're not particularly good at matching faces, although that is a thing you would think they would be very good at. They're much better at matching junk objects that we collect from the internet, and that's the kind of experiment I'm going to show you. And one really basic way of thinking about this, how an animal does a match to sample task like this, is that we have stimulus presented to the animal environment. It's going to make a response at the end of that delay period. So there's some mental event that must bridge between the stimulus and the environment during the delay period that allows the animal to identify the correct image at test, the one that it saw during study. And what I want to point out here is that there are multiple ways, at least two that I'm going to talk about, that an animal could solve this problem, or that a brain can solve this problem. And discriminating between these amount to asking some questions about what are the cognitive properties of this mental event that's occurring during the delay that allows the animal to pick the correct response. And so what we're going to talk about next is some of the properties that you might expect have to do with this mental event, or what properties it has. And we're going to do that through a series of dissociations, that is, manipulations that affect one of these systems and leave the other intact. But I want to start out by giving you some definition about what these two types of memory might act like. So the first one is, we're going to talk about it as, or we're going to call it familiarity. These are familiarity signals. They record the fact that a stimulus has been previously experienced. So sometimes they don't record much else, but that's the kind of feeling you might have when you see someone that you feel like you must know who they are, but you can't remember where you met them, but you're getting a familiarity signal nonetheless. And a couple properties of familiarity signals is that they're relatively long-term, and we have very large capacity for these kind of signals. So you can have just a vast number of stimuli to which you will have a familiarity response, given that you've experienced them previously. And what's going to become pretty relevant in the stuff that follows here is that they don't seem to require any active maintenance during the delay period. You can retain these signals without any cognitive effort. And this contrasts with working memory, which is another memory process, which again, I'm sure many of you are familiar with. But working memory is a memory process that holds specific information briefly for use or manipulation. And the properties of working memory are quite different than this familiarity signals, in that working memory is a relatively short-term process, and it has a very limited capacity. There's just not many items that we can hold in working memory at a given time. And humans at least experience working memory as active and effortful. It takes our attention, it takes cognitive effort to hold things in working memory. And we're gonna pursue that difference a little bit in monkeys here in a moment. Now, I just want to remind you, either of these, the reason I'm bringing these two up is either of these could be related to the mental event that's bridging the delay in a match to sample task. That is, animal sees a sample, they could become familiar with that sample, then at test they pick the one that's familiar. They could also see that image at the beginning of the trial and hold it actively in working memory, engaging in cognitive effort, so that it's available at test and could be used to pick out the correct image at test. And we're gonna dissociate these two memory systems, meaning that we're gonna look at some manipulations that affect one but don't affect the other. And then that is telling us that these two systems are operating in parallel. They operate somewhat independently. They represent distinct systems. And I should say, we didn't say this at the beginning, Eduardo didn't say at the beginning, I'm very happy if I'm talking past something that you want to ask a question about, just unmute yourself and give a shout. I'm not going to be able to monitor everybody on the screen, but you're welcome to ask a question. So one of the things that we found with our studies of these, did someone say something? Okay.
No, doctor, not yet.
Okay. That these that dissociates or distinguishes between these memory systems is the set of images that we use, and specifically the size of the image set that we use. And so we can drive an animal towards using familiarity or using working memory by changing the set size of images that we use for the memory test. So if we use a really large set, in fact, a trial unique set is the extreme version where they're only going to see the images once in their whole lifetime. This takes the following form: that you see a sample here, you're seeing a cow at the beginning, then you're seeing 3 pictures of animals. You got to pick the cow to get your reward. On the next trial, though, we're not using any of the stimuli that were used from this trial. We're starting with new stimuli. So now you see the rabbit, and the distractors are new. So each trial, you have a chance to become familiar with a sample and then execute a test, and you can potentially solve that test on the basis of familiarity alone. And as I suggested, we think that's probably not under cognitive control. It doesn't require effort during the delay period. You can wait until the stimuli appear, and one of them will seem familiar. So when we do this, that drives the system towards familiarity. It doesn't mean you have to use familiarity, but it makes it easy to use familiarity to identify the correct image. We contrast this with what we think of in the working memory case, where we have a small set of images. This is a kind of what you would typically have used certainly many years ago in the classic kind of pigeon and animal experiments that were done before computers became widely available. We'd have a small set of images that repeat, and we think this probably engages cognitive control, partly because, as you'll see, you look at the test like this, so we see this is exactly the same kind of test that happened in the large set case. You saw a cow. Now at test, you got to pick the cow. But on the next trial, now one of the distractors from this test is now the sample. So you've got to remember, okay, giraffe is going to be the correct response on this trial. Even though cow got rewarded last time, it's giraffe that's getting rewarded this time. So one of the things that happens here is you get a buildup of proactive interference. You can get confused which is the image you saw in the current trial. And the other thing that's happening here probably is that now all the images are familiar, because you see them every test, you see all 4 images. So familiarity becomes a really poor signal. It's not impossible to use it, because maybe the familiarity signal gets bumped up a little bit each time you see the image, but it makes it much harder to use the familiarity signal and pushes you towards a reliance on working memory, probably holding the item actively in mind. So Ben Basil was the first one in our lab to do some work on this and look at the different properties of tests with these 2 kinds of image sets. So again, we're looking at a match to sample type trial. Monkey touches this green square to start a trial. They see something to study. You have a delay period, and then you have a test where you've got to identify the image that you saw at the beginning of the trial. And based on the kind of arguments that I just gave you, or definitions about these systems, we're suggesting or coming in with the hypothesis that if familiarity is automatic and passive, it doesn't require cognitive control. And if work active and does require cognitive control, then if we do manipulations during the delay period that cause the animal to engage in a cognitive activity that might compete with holding the image in mind, retention should be impaired in the case of working memory because you're having to use cognitive control, but it wouldn't be affected in the familiarity case because you don't have to do anything during the delay to retain your familiarity signal. And so what Ben did is exactly this kind of thing, is insert different kinds of concurrent cognitive load in the delay period to assess the impact of those. So one of four things could happen in a delay period. Monkey might, this is all with monkeys, nothing would happen. It's like a standard match to sample. They would see a blue square that appeared in one of the four corners. All the animal has to do is touch that image. So it's engaged in the motor task that it's going to do in the next two tasks. A third option is it would see a picture appear randomly in one of the four corners. That picture is kind of interesting maybe to the monkey, but all it has to do is touch it. And in the most demanding concurrent cognitive load, the animal sees a categorization task. So in this case, they're identifying that this is a picture that contains a human being. So they make one of the four responses to indicate that it's a person, flower, fish, or other stimulus, or right, person, fish, flower, or bird. They make that response. That's a quite cognitively demanding, particularly for a monkey. And so the really striking pattern of results that Ben got doing this work is that we find in the, oh, these are exactly reversed. So I'm very sorry about that. I was editing this for consistency and made it inconsistent. This blue line shows you the performance of animals with in the large set case, in the familiarity case. They, no matter what they have to do during the delay interval, it has no effect on their accuracy, which remains very high. In the working memory condition, again, I'm sorry, this label is wrong. These are switched. The more the cognitive load that they have to engage with during the delay period, the worse their performance becomes. So this is evidence that monkeys are exerting cognitive control in the working memory condition, but not in the familiarity condition, because when they have to do something else that's cognitively demanding, it hurts working memory, but not familiarity. And I just want to point out this is a single dissociation, because what we've done is we've done a manipulation that is increased the concurrent cognitive load, and that affected working memory, but did not affect familiarity. So Ryan Brady came along in the lab and pushed this work a little farther in multiple ways. I'm just going to tell you about one of them, but he went on to double dissociate working memory and familiarity by now generating a manipulation that affects familiarity more than working memory. And he did this by presenting match to sample in a slightly modified format here. So during the study phase, the animal would see a picture, and it's highlighted by a little dotted perimeter. This we're calling the regular study phase. There's a delay period of, say, 5 seconds, and then a test. The animal has to perform match to sample like we've seen in the past slides. In the manipulation that he made to target familiarity, sorry, this test would be easily solved by familiarity if we're using a large set, right? You just pick the one that's familiar at the end of the test. Then what Ryan did, the clever manipulation he gave, is now on every trial, we're going to present these 4 images at test, right? Or on a given trial, we're going to present those 4 images at test. And he familiarized the monkey with those images before the test. So they would see 1, 2, 3, 4 images. They had to touch them. Then they would see all 4 of them in a row like this, and he would highlight the one that's correct, that you're going to have to pick at. Then you go to this test, and now you have to pick the rubber ducky again, but this time it's in a context where you've already seen all the images. In fact, you've seen them twice. And so now both the distractors and the target are familiar, although the monkey does know which one is the one you have to pick because it's been highlighted. So that's going to make it much harder to use familiarity and push the animal, at least that was the hypothesis, towards using working memory in these kind of tests. And that is along the lines of what, or pretty much precisely what Ryan found. So if we're using a large set of stimuli where familiarity is a strong signal, what we've got here is a regular study where you don't have to see all the distractors during study. And this is when we do see all the distractors during study. You can see that there's a really strong effect on matching performance with the large set where familiarity is the signal that animals are likely using. And there's an effect in the working memory condition, but it's of a lesser magnitude. And you might think that a problem with this comparison is that we do this wacky, weird sample phase where we, they have to see 1, 2, 3, 4 the images, and then we're seeing them, showing them in a row, and they just highlight the image that's going to be the correct one at test. So we wanted to make sure it wasn't just that format of the test that was causing the difficulty for monkeys, and particularly in the large set condition. And so Ryan did something that we called false familiarization. And in false familiarization, what we did is we did exactly the same procedure, but the 3 other images that were shown at study are not going to appear in the test as distractors. You get 3 new images at the test as distractors. And again, if we do it the traditional way, or so the way that I just showed you anyway, the typical way, where all the distractors have been familiarized, we get this huge decrement in performance. But if the distractors that are going to appear at test are novel, it doesn't affect performance at all to go through this procedure of the special study phase. So it's not the study phase that's causing, it's not the mechanics of the study phase that are causing the impairment. It's the fact that we are familiarizing the animal with the other images that are going to appear at test as distractors, so they're all familiar. That makes it hard to identify the correct one. So this is a double dissociation now, right? Because we showed that competing cognitive load has a big effect on working memory, but not on familiarity. And this familiarization phase during the study affects the familiarity test more than the working memory test. So it's really pretty strong evidence that we have 2 different memory systems in play here. And I've said more than once that we think about the working memory performance as active, and requiring cognitive effort. And I just want to show you a second example of why we come to strongly believe that that is the case, that working memory is an active process, and using the familiarity signals is more passive. And this is the work of Emily Brown, who was a graduate student in the lab, who used a phenomenon or a paradigm sometimes called directed forgetting. We tend to think it's probably more appropriately called directed remembering, but it doesn't matter that much, and it's known as the directed forgetting paradigm. And what happens in this paradigm is, again, we're using good old match to sample. So animal starts a trial by touching this green square. They see a sample, and at test they got to pick out the sample that they saw at the beginning of the trial. But what was done in this kind of directed forgetting or directed remembering paradigm is, following the sample phase and during the delay, we show the animal a stimulus that tells it that it's going to have a test of its memory follow shortly. So if it sees this blue circle, it's going to have to get its memory tested. One sort of modification that Emily did of this kind of test is that we had this remember cue come on and stay on during the whole interval. So you don't have to remember whether you're in a remember trial or not. You can see the cue is telling you you've got to remember a memory test is coming up. And we contrast this with what are called forget trials. Typically, they begin exactly the same way. So the animal sees a sample at the beginning of the trial. Then they see this so-called forget cue, in this case, the yellow triangle, that stays on during the delay. And then at test now, instead of having to remember what they saw at the beginning of the trial, they perform a discrimination that they've learned very well outside of this particular paradigm. So in this case, whenever they see the pelican, they've got to select that and avoid these other stimuli. And to prevent the animal from, once it sees this triangle, start thinking about the pelican, forget trial, Emily did what I thought was a pretty nice manipulation. She gave them, I forget what the exact number is, but there was quite a few of these discriminations that they learned. So they didn't know which discrimination was going to come up, and that really discouraged them from prospectively coding the correct response. The idea here is, once you've seen the forget cue, I don't need to remember this, because I'm going to get a discrimination test instead of a matching test. Whenever I see the blue circle, I need to remember because I'm going to get a test of my memory for that. And of course, being the evil, tricky psychologists we are, we sometimes switch that the test that follows these stimuli to ask whether they actually have the property that we think they are. So if this blue stimulus is actually directing the animal to remember, then they should do better on a test following this cue than a cue that tells them to forget it, don't worry about it. And then suddenly you give them a memory test and they've already relaxed and stopped holding it in mind. The point here is that if the animal, if it actually involves active maintenance to do the working memory task, then these cues should get control of that process. The blue cue should keep the animal in that active cognitive state, and the yellow triangle should allow it to stop being in that state. And so you'd predict different accuracies under the two conditions when we switch the probe trials, as Emily did. And the interesting manipulation that she did here is that she did this both with the working memory type stimuli, where we have stimuli that repeat, right? And we expect the animal is using working memory. And you can see the results here that on regular trials, where shown in the dashed line here, where the animal gets the remember cue and then gets the test, and the lower line where their performance is worse is the trials where the monkey got a forget cue, so don't worry about it, you don't need to remember that, but in fact they got a memory test. So that's consistent with active working memory, cognitively demanding active working memory. And then she also tested them with a large set of trial unique stimuli. And what we found under those conditions, there's no difference between the remember cue and the forget cue, suggesting or consistent with this idea that memory for the sample in these working memory tests is under active cognitive control, and there's nothing to control about familiarity. It just sits there waiting for the test to appear. Okay, so that's really the first part of the talk that I wanted to share with you, just setting up that there are these two memory systems we're pretty confident are happening in rhesus monkeys. And then sort of armed with these or prepared with these two memory systems, we want to do some comparative psychology and ask whether there's differences between animals in the exercise of these two memory systems. And we start out with a particular monkey-ape comparison I'm going to tell you about today. As you would know, it's involving orangutans. And I just wanted to provide a little bit of motivation for why we do this. You know, the apes, including orangutans, although orangutans are a little more distantly related to us than other apes, are more closely related to humans than monkeys. So we're able to look at the gradient of ancestry here a little bit. And we know in the apes from work, including work that I see Jem Rilling is here, some work that Jem did, we know that the prefrontal cortex of apes is proportionally larger than it is in monkeys. And through a relationship that we have with Zoo Atlanta, which we're really grateful in the lab to have this opportunity to work with their collection of orangutans and also gorillas, although I won't be talking about gorillas today. We really appreciate that we are able to do some of this work at Zoo Atlanta. It's definitely an effort for them to make space for us to do it. We think it's a great thing, though. We get to learn things about apes that we couldn't possibly learn because we don't have apes in laboratories. And it's probably also a good thing for the animals in the collection that they get to engage in some stimulating computer activities for part of their day. Now, a huge problem that we have in trying to compare the apes at the zoo and the monkeys in our lab is that they could differ in any number of different ways. There could be a whole lot of things that are different about their life experience, their experience using computers, and how they were raised and so on. So we need some way of trying to address that we don't want to or we almost certainly don't want to look at just raw accuracy scores. It needs to be sort of somehow scaled to ask about these different memory systems. And so roughly the way we came up to do this, given that we'd worked out working memory and familiarity, and we thought, well, the apes have a large prefrontal cortex, perhaps they're going to also have a particularly pronounced working memory, so they might be a little more likely to use working memory than would a monkey. And if we can get measures of both these kinds of memory, both the working memory and the familiarity memory, and if familiarity memory is, one naively might think is sort of an ancestral kind of memory that would be very common across species, this ability to recognize something has been previously experienced, very close to habituation, in fact, that you have a different response to a stimulus on the second presentation and third presentation. So if we could express working memory performance as a fraction of familiarity performance, that might be a way of calibrating for whatever other differences exist between the animals, just in their familiarity with cognitive testing and so on. So that is the logic we went into this work with, and you'll see that logic maybe didn't obtain or didn't follow through the way we expected, because that brings us to the mystery of the missing memories in orangutans. So this is work done by Ryan Brady, who's a graduate of the lab and who's spent a bunch of time at Zoo Atlanta with the orangutans there. And so now what I'm going to do is just show you a few findings, comparative findings, where we directly compared orangutans and rhesus monkeys, and got presented with some puzzles. So the first puzzling finding that we had with the orangutans is that they don't show a difference in memory accuracy with set size, the one that is very well established in rhesus monkeys, that when they're working with a large set of novel images, their performance is better than when it's them working with repeating images. So that's the kind of finding you see here with 6 monkeys. If we give them a large set of non-repeating images, or we test them with repeating images where proactive interference is high and familiarity is shared by all the images, the monkeys just do better on the trial unique tests than on the repeating images test. Again, these are the ones that where familiarity would be helpful, and the repeating ones are where you have to use your working memory. In the orangutans, we found no difference between those. So that by itself was already sort of a puzzle, like, ooh, what's going on there? Why aren't they getting a benefit from familiarity? But it's consistent with the idea, if we express these as a ratio of each other, well, in monkeys, maybe working memory is not so potent because they have a smaller frontal lobe, and the orangutans maybe are more prone to use working memory because of their larger frontal lobe. The difference between these two kinds of tests is smaller, non-existent in orangutans, but working memory is less pronounced in the rhesus monkeys. That's possible. We did expect to see some difference here. But so we proceeded on to a second experiment to test more directly what are the orangutans doing in these tests with either large or small sets of stimuli. And that brought us to our second puzzling result. So on the left, we have monkeys who are getting that test that I showed you. This is a replication of that study where we give them a concurrent cognitive load during the delay period, so that they, if they're actively holding something in mind, that concurrent task is likely to bump that memory out of mind. We see with the monkeys again, if the stimuli are trial unique, that is, each test is new, the distractors are novel. Having them do this competing cognitive load that's shown on the x-axis, either nothing happens or they have to go all the way to make a categorization response. No effect in the familiarity. The tests we think are engaging familiarity, and a huge decrease in performance with increasing cognitive load if they are using working memory. The orangutans, by contrast, show a competing cognitive load effect in both cases. This now is really pushing us towards the idea that even with trial unique images they've never seen before, and tests that are going to involve novel distractors, the orangutans are using their working memory. That's how they're solving the task, because if we ask them to do something else during the delay period, it actually impairs their performance. So that was the second piece of the puzzle that we're just thinking, well, why? We thought this familiarity signal was probably shared by most species. When we go look now, we're seeing not only are they not using the signal, but they seem to be using working memory exclusively, even in conditions where monkeys readily use that familiarity signal. So we thought, well, maybe maybe the orangutans are just, their frontal lobes are so big and they're so good at doing working memory, they just default to that all the time. Let's give them a test that you can't possibly solve using working memory, and see if then the reliance on a familiarity signal will come out. So Ryan developed this, excuse me, this novel task that can only be solved by familiarity. At least we don't think it can be solved by working memory. And in this test, what happens is the trial for monkey and orangutan starts again by touching a green square, as it usually does. And then we present them with a whole series of images that are appearing randomly in the corners of the screen. Each time they see one, they have to touch it. The idea is they're becoming familiar with these images during this phase. Every fourth image is followed by just a really simple discrimination test, where you got to find the square that contains the circle with the smaller line. And that's so we can give them a little reward every once in a while, so they'll keep poking their way through the images, because we show them 40 images this way. So they're going to get 10 cycles of this. And in fact, we repeat it so that they have multiple exposures to each one of these pictures, with the intention that they're becoming familiar with them. This can take 10, 15, 20 minutes. And then after that test, or after the study phase, then we start presenting them with a series of tests now, where each test is going to involve presentation of one of the items that they saw during the familiarization phase, and 3 distractors that they've never seen in their life. So these are images they've just never, ever seen. So you see here, you got banana, you got to pick that. This one, you got to pick grapes, because grapes appeared during the study phase, but the distractors did not appear during the study phase. They'd never seen them. So because this, first of all, it takes at least 10 minutes for them to get through this study phase, and because we're presenting 40 images, we think we've just overwhelmingly exceeded both the duration and the capacity of working memory. So you really need to solve this problem using familiarity, that is, identifying the stimulus you've seen previously, even though you haven't been holding it in mind. And so we've got comparative results on that, comparing the monkeys and the orangutans. So this is a little bit unusual presentation of the data. What we did is we asked the monkeys, we asked the animals to achieve 70% accuracy, where 25% is chance in these tests. And then we gave them 10 sessions to meet that criterion. If they didn't meet the criterion, we gave them a shorter list that would be easier. So we went from 40 to 20 to 8, and finally to 1. So let me show you the monkey data first. So out of 10 monkeys that we tested, 7 of the monkeys immediately met the criterion in the first 2 sessions. That's the fastest you could possibly meet criterion. They didn't have any problem picking the image they'd seen before. Of the 3 monkeys that didn't do it in the first 10 sessions with these tests, 2 of them did it with the 20 list, and our slowest monkey finally acquired it with a list of 8 images preceding the series of tests. Now, giving the exact same thing to the orangutans, what we see is out of the 40, none of the 6 animals that we could test were able to do it. We give them 20, a list of 20, none of them can do it. You give them a list of 8. None of them meet criterion on this simple test where you just pick out the image you've seen before. And you might think at this point, well, what is the orangutan doing? Maybe they're thinking about this all wrong. They're doing something else. They don't realize it's a memory test. But when we reduce the list to a length of 1, all of a sudden their performance jumps up. So now this is the first time that probably working memory is a viable strategy, because you don't have so many images and so much time passing before the test. But there's a funny thing embedded in this. Remember, I showed you that each time they see an image, then they have to do this test where they find the small target and touch that before they can proceed to the test phase. That's a concurrent cognitive load. We see the sample, then you got to find that small target, and then you get the test. And if we take away that concurrent cognitive load, the performance jumps up even higher, almost to the criterion. So we're pretty sure this whole time the orangutans knew they're in tests that involve match to sample, but they simply are not tapping into that familiarity signal. And only once you start, you return it to a condition where you can solve it as a working memory task, do you see accurate memory performance. So boy, has this ever left us puzzled. We thought this was kind of probably an ancient, ancestral, widely shared kind of memory, this familiarity signal. It really is close to the kind of signal that you think would underlie habituation, which does involve kind of memory, right? You've seen the stimulus before. Second time you see it, your response would diminish and continue to diminish with repeated presentations. And we just can't believe, I think with very good reason, that orangutans don't habituate at all. They clearly do. But somehow they're not tapping into that signal with the kind of tests we're giving them, and we're really stuck about why that is, or puzzled by why it is. Nonetheless, we thought, well, we should maybe take this closer to a habituation paradigm to sort of parameterize where is it that they can use these signals and when is it that they can't use these signals. And so Johnny Engelberg and Mackenzie Webster both started doing some tests. These are postdocs in the lab. Johnny has moved on. Mackenzie's still with us doing great work. Presented the orangutans and the monkeys both with very simple kind of discrimination test where each trial you touch the green square to start it, and then you got to pick the smaller square at test. And so we can pretty readily train the animals to do this, pick the smaller square. But then the way of measuring whether they're habituating to stimuli they've seen is that we then sometimes present these squares as images, and those images can either repeat, providing the opportunity for the animal to habituate to the picture and then focus on, I just got to find the small one, just find the small one, don't worry about what's in the picture. Or they can be trial unique, that is that each time they get a test, it's a new image, and if you aren't habituated to that picture, you might get interested in the picture. That's going to distract you and slow down your finding the small target. At least so the thinking goes. And so we have blank trials where there's no image. We have images that repeat, that the animal might habituate to and show quicker performance with the repetitions. And then we have trials where the images are a new image they've never seen before, and they might be interested in that image, and that might slow down the target finding performance. So indeed, when we do these tests with monkeys, what you're seeing here is the two species we have. Then we're the response latency, finding the smaller target. And then we have, if it's a blank squares, they're quite quick. If it's images that they're habituated to, that is, they've seen them multiple times now, they're intermediate speed in finding that small target. And if they're novel, well, the kind of thing we expected. They're interested in the image maybe, or the image is interfering with their ability to find the smallest one, and they show the slowest response pattern there. Now, the orangutans, they show the same stacking pattern, but the difference is definitely muted, and it's not significant as a function of whether they're habituated or novel stimuli. We do get a significant difference that they do better when there's no picture to look at all. And that turns out to be important because this is not the only experiment like this we've done. We've done several of these kind of target finding experiments to look at habituation. And one version of them, instead of having the target appear inside the images they're trying to find the smallest one of, it appeared in the middle of the screen, and we thought, well, if they just see a new image in the middle of the screen, it's going to draw their attention unless they're habituated to it, and that's going to slow them down. And in fact, that's what it did with the monkeys. It didn't do that with the orangutans, but we thought, well, maybe the orangutans are just really good at ignoring this distractor that's in the middle of the screen in an area where they don't have to make their response. And so we embedded the stimulus in the stimuli to which they have to respond, and we're seeing the same effect again. So we've done multiple versions of this that are more or less showing us the same thing, and Mackenzie's wrapping up a final experiment that I'm not going to share with you because we don't have the full data here. But pretty surprising to us that we're not even in this, what we think is a super simple paradigm that ought to measure habituation, we're not seeing a really clear habituation signal in the orangutans, at least a much attenuated one. So those are the data that I have to share with you, and I'll just offer you a few tentative conclusions here. The ones in monkeys won't be entirely tentative. I think they're pretty solid. The ones in monkeys, we know that there's both working memory and familiarity systems in the monkey, because we can dissociate them behaviorally. We can manipulate set size. We can manipulate concurrent cognition. We can use the directed paradigm, and we see these consistent patterns of difference in the use of those two memory systems. So we think they're there and they're distinct. If the distractors are novel, we see a really powerful familiarity response in the monkeys, and that protects them from competing cognitive load. When you can't use relative familiarity, then and working memory must be engaged, then it becomes more effortful and more cognitively demanding. Now, the comparative conclusions, I want to be clear, probably ought to be a little more tentative because we're so puzzled by these. But we find that the orangutans are more likely to use working memory, and that's consistent with the way we went into this work, with the sort of naive working hypothesis we had that because they have a large frontal lobe, maybe they're going to be more likely to use executive function. They're going to use working memory. So that is consistent with that view, but it's still just kind of shocking and surprising to us that we're just finding no evidence whatsoever of a familiarity signal, even when we designed a test where that's the only solution that the orangutans could use to solve the problem. Even to the point that we did this habituation paradigm, and we're finding very little, if any, evidence of habituation under those conditions. So we're left with this struggle to figure out what's going on here. Do the orangutans genuinely somehow lack a familiarity signal that rhesus monkeys, and by the way, human beings, seem to show quite readily? Somehow, almost certainly limited maybe to the kind of images that we're using in these computer tests somehow. It's also possible, it still remains a little bit of a possibility, I think, that these the animals just have a kind of extreme cognitive control, so they're able to ignore the distracting stimuli in the discrimination test, where they got to pick the stimulus that's smallest, and maybe are just ready to do working memory with novel stimuli, even when they wouldn't have to do that. One reason you might be a little cautious about this idea that they just have extreme cognitive control, and that's why we're not seeing familiarities, because they're just overwhelming it with cognitive control all the time, is they really show pretty pronounced effect to concurrent cognitive load in a working memory task, right? We still see that same decrement in performance as we increase concurrent cognitive demand with the orangutans as well. So I'll close with some really speculative neurobiology about these orangutans. So some frontal lobe comparisons that have been done with post-mortem orangutan tissue report that they have a small ventromedial prefrontal cortex relative to other apes, and this is an area that has pronounced connection to the perirhinal cortex in a primate brain, and that connection is one that probably drives the familiarity response. We know that damage to this area in monkeys attenuates performance with a large set. I feel very cautious about, we don't know a lot about this neurobiological difference, and we certainly didn't go into this work with a hypothesis about the neurobiology, but I think it does stand as a possibility that that's some part of the puzzle here, that they do somehow lack the neurobiological mechanism that's primarily responsible for this familiarity response. And before I pause for questions, I hope you don't mind a little advertisement. We are looking for a postdoc in the lab to do some work on auditory cognition and auditory cognitive control, using some of these kind of paradigms that I've talked about, and other ones that we typically use in the lab to do with metacognition, to look at differences between auditory processing and visual processing in monkeys. And so we're very eager to hire a postdoc and or a research coordinator for these positions. And if you know anybody who'd be interested, these are multi-year NIH funded positions. So it'd be a great opportunity to get involved in some primate work if you're suitably prepared for that. And with that, I will just close with a little reminder slide of some of the things that happened, and I'd be happy to take any questions if anyone has any. Thanks for your attention.